Right. Well, it's 12:30, so let's get started. So, as you hopefully know, your exam is next Thursday in this room at this time. So make sure you come prepared for an exam next Thursday. Bring a pencil, bring a calculator. In terms of the actual exam, it'll be like on paper. I'll print it out for you so you can write all your work and stuff on the exam itself, so I can check your work and give partial credit for calculations and things like that. I guess maybe now's a good time to look at on the bright space. So this is really small, but I posted for next week extended office hours. So I'll have my normal office hours on Monday next week. And if you want to see me anytime on Tuesday, just let me know. We'll make a Zoom meeting. And on Wednesday, I extended my office hours so they go until 3 p.m. in case anyone has any last minute questions. I will not be having office hours Thursday after the exam because I don't think anyone will want to talk to me after the exam. So, but if that's different, you can also email me and make an appointment. I also posted the reference sheet that you'll be getting with the exam so you can see exactly what reference materials you'll be provided with. I also posted review questions for the exam. That's a mixture of review from chapters 1 through 5 that we've been going over. And this coming Monday's class, right before the exam, we will finish up chapter 5 if we don't finish it today. And then if anyone has any general review questions about anything from chapters 1 through 5, that's the time to ask if you want to ask it in the class. I don't have a specific review session I'm doing with you on Monday, so if you want to review anything, you bring the questions. So that is what I wanted to say about that. So let's now finish or keep going with chapter 5. So if I recall, this is where we finished on Monday, talking about protein expression using the lac operon as our little inducer of our protein A. And so the next, I think I actually also showed you the little animation, which is from the slides about how when the lac repressor is bound to the operator, RNA polymerase is unable to transcribe your gene of interest, so you don't get it. Then when you add IPTG binds the lac repressor and pulls it off the operator, so then RNA polymerase is able to transcribe your gene. And then once the gene is transcribed, you can translate it into the protein that you want. So once you make your protein, the next step is to purify it. So let's go over some different protein purification techniques. So everybody take a couple of minutes and think about these 6 protein purification techniques and then what each of them does. And then we'll go over it together.
I have a quick question for you. Why don't you just use lactose instead of FKTG? What's the difference?
So you can use lactose or allolactose, but it's so like the I was going to say, like, if you have beta-gal, then beta-gal will cleave it, and then you would eventually no longer have lactose. I mean, the idea with IPTG basically is it's not cleavable. It's more stable in solution, so it will last a really long time, as opposed to using something like lactose or allolactose that'll break down quicker, so you might not get as high of an expression level. All right, it looks like at least some of you have stopped writing. So let's start going over these. So who can tell me, for the first one, which of these techniques separates proteins by their size? Yeah. Yeah, excellent. So gel filtration chromatography helps to separate proteins by their size. Another name for gel filtration chromatography is size exclusion chromatography. So you might see that sometimes. But if I wrote that in the clues, that would be a very obvious clue that it separates proteins by their size. So sometimes it's also called gel filtration. Two names, same thing. Okay, so number 2, which one separates proteins by their solubility? Yeah, salting out. Great. So we can use salting out. That's a really common technique for first removing proteins that are less soluble than your protein of interest, and then other way around. First removing proteins that are more soluble than your protein of interest, and then removing your protein from the proteins that are... I don't even know what I just said. I confused myself. So first, let's start again. First, you remove proteins that are less soluble than your protein of interest. Then you remove your protein of interest from the proteins that are more soluble than it is. And somewhere in here, there's a diagram of that when we go over a question with it. Okay, so number 3, which one of these separates proteins by their binding specificity? Yeah, affinity chromatography. So affinity chromatography is a really helpful technique because it can be very specific to just take out your protein of interest from all the other proteins in a mixture. But the downside is your protein needs that specific thing that will bind to the column. So not all proteins will naturally have some kind of specific binding affinity, but you can often genetically engineer proteins to have, for example, a histidine tag, which we talked about in the lecture. All right, so number 4, which one separates solids from liquids? Yeah. Yeah, so centrifugation. Centrifugation, you take a tube with a mixture of things, you spin it really fast. More dense things and solids will pellet at the bottom. More soluble things and tinier things will stay suspended in the supernatant. Okay, so number 5, which one separates proteins by their charge? Yeah. Yes. Ion exchange chromatography separates proteins by their charge. And we'll do another practice problem more specifically about that coming up also to get more familiar with how that works. All right, so the last one, which one of these breaks up cells to release cellular proteins? Yeah, homogenization, exactly. So homogenization is a technique. It can be as simple as putting tissue in a blender or using something a little fancier like sonication to break cells open and release the contents of the cells. So if you, like we were just talking about in that previous example, if you had a bacteria that you genetically engineered to overexpress a protein, which one of these techniques would you do as the very first step for purifying your protein? Yeah. Yeah, exactly. So you always want to start by homogenizing your tissue or lysing your cells, because otherwise your protein will be hiding inside of a cell. You can't get it out, right? So always start here, and then a really common next step after that is salting out. Salting out is a really good way to just separate out proteins sort of in bulk based on their solubility. So a lot of times your first step is going to start with homogenization and then salting out. And then from there you can choose to do a different column chromatography technique, sort of depending on the properties of your protein of interest, which one you'll pick the best one depending on what your protein is and the properties of your protein. And a step like centrifugation is a step that happens multiple times throughout the process. So you might do a first initial centrifuge right after you homogenize your tissue, just to kind of separate out bulk solids from soluble things. And then each time you do a purification step, you'll often do a centrifugation to separate out one piece from another piece. So, all right, so let's start going through these steps. So let's start with homogenization, because that's what you do first. So which of the two of the following techniques are types of homogenization that are used in protein purification? Yeah. Yeah, so sonication is one, blending is another. So yeah, I just mentioned those on the previous slide. So sonication is when we use essentially sound waves to vibrate cells and destabilize their membranes and cause them to open up. Blending is just simply using the sharp blades in a blender to physically break open cells or tissue. These other things here are also different things you can do to solutions with cells in them, but they don't necessarily homogenize the cells or cut them open. Any questions about homogenization? All right. So next thing after you do homogenization is often salting out. So here there's a table that has 3 different proteins that we'll use for the next couple of examples with the different properties of the proteins, including size, solubility, their charge, and any particular domains that they might contain. So when you want to do salting out, when you progressively add more ammonium sulfate to a solution, what order are these proteins going to precipitate in as you keep adding ammonium sulfate? So everybody take a couple minutes and think about that, and then we will go over it together. All right, so which one of the properties of a protein do we need to look at when we're talking about salting out? Yeah, solubility, exactly. So when we're looking at solubility, which protein will precipitate first as you keep adding ammonium sulfate? Yeah. Yeah, exactly. So if you add a little bit of ammonium sulfate, first you'll get the lowest solubility protein will come out first, and then what would come out next? Yeah, and then next the medium solubility protein, and then finally when you add a high enough amount of ammonium sulfate, the highest, most soluble protein would finally come out of solution and precipitate. So just as a little review of how salting out works, I have the slides from the lecture here. So if you have a tube with a mixture of a variety of proteins, and here there's yellow ones, red ones, and green ones, and your protein of interest is green. So first you'll add a low concentration of ammonium sulfate. So that would precipitate out these red proteins, which are not very soluble. And then the green and yellow proteins are more soluble, they'll still stay in solution. As you keep adding ammonium sulfate, then you'd precipitate out the green protein, which is your protein of interest, and it would leave the more soluble yellow protein still in the supernatant. So then from there, you could take this pellet that contains your protein of interest, add more buffer to it to suspend it again, and then keep purifying from there. The next step you would usually do once you've isolated your target protein is you'd use dialysis, and that would help you remove all of that ammonium sulfate salt from your protein, because a lot of times you don't want all the salt in your final product as you continue to purify. Any questions about salting out?
Can you explain how the dialysis procedure with the ammonium sulfate?
Sure, yeah. So dialysis, as you know, has a semi-permeable membrane that lets water and small molecules pass freely through. So if your protein of interest is, let me draw a picture. So here's your dialysis tubing, your protein of interest is inside, and then your ammonium sulfate, I'll make that purple, is also in here. So the ammonium sulfate can freely pass through the membrane into the solution. Your protein stays trapped inside. So then over time, eventually the salts and other small molecules can exit the dialysis membrane, and larger molecules such as your protein and other large contaminated proteins would stay inside the dialysis membrane. Does that make sense for everyone?
Yeah, for some reason I thought like the ammonium sulfate actually like attached itself to the protein, not actually causing precipitate out. So I can see
Right.
ammonium sulfate's really.
Yeah, I mean, when it's in the pellet, certainly they're attached, but you would take this pellet and then resuspend it in more buffer, and then the ammonium sulfate would go back, everything would go back into solution, because you're essentially diluting out the ammonium sulfate in like your new solution. Yeah.
Wait, I don't understand this before, but how would you then remove like the red protein so that you could just do allow the green ones?
Oh yeah, so after the second step, like when you're here.
Yeah.
So at that point you can pour off or pipette out the supernate, which contains your protein of interest and other very soluble proteins, and you would leave the target protein as a pellet in this tube. Put everything in like a brand new clean tube, and you'd only be bringing the green and the yellow proteins with you. Any other questions? All right. So next, let's start talking about different types of column chromatography. So first of all, for any type of column chromatography, can somebody tell me what the stationary phase is? Like a definition for the stationary phase.
Maybe like what's interacting with the protein as it goes through the column.
Yeah, exactly. So the stationary phase stays in the column. And it interacts with proteins. It's often some kind of gel or beads. And sometimes it's called the resin. So you might see any of those terms when you're talking about the stationary phase. So if you know what the stationary phase is, what is the mobile phase? Yeah. Exactly, yeah. So your mobile phase is a solution that contains your protein of interest, the POI, and other contaminants. It's usually a buffer. And it, right, as you said, it passes through the stationary phase, or it passes through the column. Any questions about, yeah?
So on, in the video, when you were talking about the stationary mobile phase, there was also like stuff about the different forces that you can use to push through.
Yeah.
On that slide, you mentioned something about how there's something called a preparatory separation method.
Oh, so what I meant by that was just so like there are basically two things you can do when you're purifying a protein. You can make a large amount of it that you're going to do something with, and that's often called like preparative or preparatory because you're preparing it to be used, versus like an analytical usage where you just use like a really tiny amount that you can analyze, but you can't like take it and do something with it later. Sorry if that terminology was confusing. Yeah. Yeah. Yep. Any other questions? Yeah.
So I know for like the ion exchange chromatography,
Mm-hmm.
our sample mixture that we pour into the stationary phase is considered a mobile phase, but is the buffer that also passes through considered a mobile phase?
Yeah, like everything, the buffer and all the stuff in it is the mobile phase. Yeah.
The low salt elution buffer is also considered the mobile phase?
Yeah. Yeah, anything that you pass through the stationary phase, whether it's buffer containing your sample or a washing type buffer or an elution type buffer, is all different types of mobile phases you can use. Yeah. Right, usually a buffer. All right. Any more general questions about columns? Okay, so let's do some more example problems using the different types of chromatography. So this next question is about using gel filtration chromatography. It's the same table of proteins that we saw with the other problem. So now I want to know, in which order are these proteins going to elute from a gel filtration column? So everybody take a couple of minutes and think about that. All right, so which property of the protein do we want to use here for our gel filtration column? Yeah.
The size?
The size, yeah. Which one would come out first? The smallest one. Okay, so does anybody agree with that? Is it A, the smallest one that comes out first?
It should be C because the small ones get stuck in the actual beads.
Yeah, exactly. So with gel filtration chromatography, it's actually the largest proteins that come out first. So in this case, protein B would elute first, followed by the medium one, which is protein B, and then the smallest one would actually come out last. So this is a little bit opposite of when you're thinking about, for example, like gel electrophoresis, right? In gel electrophoresis, the smallest molecules, either DNA or protein, depending what kind of gel you're running, the smallest ones will travel the furthest, the fastest. For gel filtration chromatography, we have the opposite. It's the large proteins that travel the fastest. The small proteins, like you said, they get stuck. So let's look at a picture to get a better idea of what that means or how that happens. So gel filtration chromatography is the stationary phase is made of these gel beads that have a gel matrix inside of them that has basically little pores that run through these beads. So really small molecules and really small proteins will be able to enter inside the beads, and they kind of get trapped in there. They have to pass through the maze and take a really long route to finally get to the other side of the bead. Versus a larger protein, which is shown here in blue, it can't fit inside the bead, right? It can just go kind of straight around. So it has a much shorter path to travel, so it's able to move quicker. Basically, it comes out first versus the smaller ones that get trapped. So when we look at that from our little summary, so you have, for gel filtration chromatography, you have these gel beads that are your stationary phase, and then you have your solvent containing your proteins. And then over time, the larger proteins, they'll be able to travel first, they go faster, versus the red ones that get kind of trapped towards the top because they're small and they're stuck. So eventually your large blue proteins elute first, and then your tiny red proteins come out later. And if you collect in different fractions, you'll be able to, in different tubes, right, different fractions in different tubes, you'll be able to separate out which tubes have your blue protein and which tubes have your red protein. Any questions about gel filtration chromatography? Okay. So next we have an ion exchange chromatography question. So this is talking about cation exchange chromatography. And so the question, same three proteins, in which order will these proteins elute from a cation exchange column with increasing eluent strength? So everybody take a couple of minutes and think about that. Okay, so for cation exchange chromatography, which property of the protein are we going to use to separate? Yeah, the charge. Yep, the net charge. So for cation exchange, does the stationary phase of the column bind to positively charged or negatively charged proteins? Yeah. Yeah, exactly. So if it's cation exchange, it's going to bind to cations. And anions will pass through. So for anion exchange, it would be the opposite, right? Anion exchange chromatography is sticking to negatively charged proteins, anionic proteins. Cation exchange chromatography is really good for separating out cationic proteins with a positive charge. It's not good for separating out anionic proteins because they don't interact with the stationary phase of the column. All of the anionic proteins would essentially pass through the column. So if all of your proteins are negatively charged, probably don't want to use cation exchange chromatography because it won't separate them. So that being said, which protein would elute first from this cation exchange column? Yeah. A, yeah, so the negatively charged protein, like we just said, doesn't interact with this column, the stationary phase, so passes right through. Which one would come out next? Yeah. Yeah, B. So that one has a slightly more positive charge. It will interact with the column, but not super duper strongly, so it will be the first of the positively charged proteins to elute off the column as you increase element strength. So that leaves our third one as protein C. Protein C has the most positive charge. It's going to interact the most strongly with the stationary phase of the column. It will come out last. So let's look at a picture of what that looks like just to remind ourselves. So this is a more general picture for ion exchange from the lecture. So you start with your sample mixture that you load on. Your stationary phase is an ion exchanger, either cationic or anionic, and you start with a low salt buffer. So as your sample passes through the column, the proteins will separate out based on their charge. So in this picture, first this red protein is coming out with the low salt buffer, and then as you continue to put more and more salt into the elution buffer, that will start pushing out the proteins that have bound more strongly to the column. So essentially the extra concentration of salt helps to sort of push the proteins off the column by interfering with those charge-charge interactions between the protein and the column. So yeah, so then once you add your high salt buffer, then your slightly more sticky protein comes off, and then your most sticky yellow protein comes off last. Any questions about ion exchange?
I'm confused about what fraction number for one element means. Like, why would it be low for the first protein that comes out?
So this is the fraction number just means so as you are running buffer through your column, you're collecting it in these little tubes. This is showing test tubes. You can use whatever tube you want. And generally we'll just call each successive tube a fraction. So the earlier fractions, like number 1, number 2, number 3, your less sticky proteins that come out first will come off in the earlier fractions. And then the 50th tube you collect might have the most sticky protein that comes off last. Yeah.
So if you have how do you choose which whether to do an anionic or cationic column based on the protein count? Like, do you want it to be one of the first fractions that you're collecting, or do you want it to be one of the later fractions that you're collecting? It doesn't matter.
I would say it depends a lot on the properties of the protein itself, as well as what other proteins are in the mixture of proteins. So you want to set up the chromatography in such a way that it will separate out your protein of interest from whatever else is in there. So like if your protein has a positive charge at a certain pH, and all the other proteins in the mixture will have a negative charge at that pH, then you'll want to use cation exchange. That way your protein will stick to the column, everything else will come straight through, and then you can collect your protein last all by itself. But if your protein happens to be negatively charged and it's in a mixture with positively charged proteins, then you'd want to do anion exchange. The answer is it depends, I guess. Yeah.
Could you do it the other way?
Yeah.
Your protein of interest comes out first.
Sure, yeah, absolutely. It sort of depends what you want to do. So if you care about kind of separating out all of the individual proteins in a mixture, then that would make sort of more sense. If your protein's like the only positive one, the other ones will stick to the anion exchange. First your protein comes out, and then you can separate out all those other negatively charged ones as you elute them one by one. It just sort of depends on your goal for the end of the purification. Any other questions about ion exchange chromatography? All right. So now we have affinity chromatography. Same 3 proteins. In which order will these proteins below elute from an affinity column containing a nickel resin? So take a couple minutes and then we will go over it together. Alright, so which property of these proteins are we going to look at for affinity chromatography? Yeah. Yeah, this column where the special things that the protein contains. I guess there's not really a good word for that. Binding partner possibilities, I guess you could call that. So if you have an affinity column with nickel, which protein will come off first? Or maybe I should flip it around and say which protein will stick to the nickel resin? Yeah. I'm sorry? Yeah, exactly. So this his tag protein is a really strong binding partner for nickel. So this protein B is going to stick to the column. It's going to come off last, right? So it's number 3. Then we have these other 2 proteins, right? A and C. They don't have a his tag. So, yeah. So, yeah, actually in the case of affinity chromatography, the only thing that matters is the binding partner. So these 2 other proteins would actually come off pretty much at the same time. So they're tied for number 1. They don't interact with the column. They're going to travel through it at around the same speed. So affinity chromatography is really powerful for this reason, because it can bind really strongly to your protein of interest and not interact almost at all with other proteins. But the tricky thing about it is not all proteins are going to have some kind of binding partner that's naturally part of the protein. So those of you who are in biochem lab might know an example of a protein that naturally has a binding partner. So we've been isolating LDH in the lab, which interacts with NADH. So we used affinity chromatography this week to base it with a stationary phase that mimics NADH, and that allowed LDH to stick really strongly to the column and allow other contaminant proteins to pass through. In the case of a histidine tag, most natural proteins are not going to just have 6 histidines in a row. So usually you only get one of those if you genetically engineer the tag into your protein. But whether it's genetically engineered or natural, if you have some kind of binding partner, affinity chromatography is often a purification technique that you'll go to. So let's just review affinity chromatography real quick. So first we have our protein, and we have in this example, we have our histidine tag, so it's 6 histidine residues in a row. The lone pair on the nitrogen will bind really strongly with the nickel that's attached covalently to the actual resin of the column, like the bead. That's the blue circle, that's the actual sort of beads of your column. So if you have this sort of large picture of it, so you have inside your column, you have your solid resin matrix with your matrix anchored ligand on it, which is sort of this part, the nickel and the resin, and then your protein of interest, which is going to bind to the anchored ligand. So in that case, it's our his tag on the protein that binds to the nickel ligand, and then there will be various ligand binding sites in your sample mixture. So as you pass them through the column, your protein, which is shown in red here, is going to stick really strongly to the matrix anchored ligand. The other proteins will pass through, so you'll collect them in the earlier fractions. And then as you keep collecting and you add an element, then you will collect your protein of interest once you push it off the column. Okay, so are there any questions about affinity chromatography? All right. Yeah, no worries.
So how do you remove the ligand that binds to protein sort of the last?
Like, how do you actually elute off your protein, is what you're asking?
Like, because the ones that come off last,
Yeah.
so that test tube, they were bound to
The nickel, yeah.
Yeah. How do you remove that?
Yeah, so for that you put something in your element that either mimics the nickel or mimics the binding partner of the nickel. So in the case of a his tag specifically, you can add just like nickel, like a solution of nickel, and that will start competing to bind with the histidine tag on the protein and eventually push it all off. The other thing you can do is add a higher concentration of imidazole. Imidazole is like this part of the histidine amino acid residue is an imidazole ring. So if you add a bunch of imidazole, that will compete for the binding sites on the nickel in the column, and then that will help to sort of push your protein off the nickel because the imidazole wants to stick to the column itself. So those are two things you can use, but they're specific to the ligand that you're using. So like that's what you'd use for nickel. If you had a different type of affinity column, you'd use something else to push your protein off the column. All right. Any other questions about chromatography? So that brings us to the next part of chapter 5, which is about assessing the purity of proteins. So first, let's take a couple of minutes and look at these different types of assessing techniques and match them to what their function is, why we use them. So everybody take a couple of minutes and try to match the technique to what it's doing. Okay, so can anybody tell me, for number 1, which technique do we use to visually assess the success of our purification steps? Yeah. Yeah, excellent. So gel electrophoresis is a really good method to visually separate out all the different proteins in a mixture and see what's there and look at their size. So if you just went through a whole series of protein purification steps, if you then go and do gel electrophoresis on your final sample, you would hope to only see one protein in there. If you have a mixture of different proteins, clearly your purification did not go as well as you thought. Okay, so for number 2, which one of these helps determine the concentration of a protein by measuring the light absorption by the protein? Yeah. D, yeah, so UV-Vis spectrophotometry. So you can use the property that proteins have of absorbing light at a particular wavelength to then see how much protein you have in your sample. And we'll do an example of that in a little bit. So number 3, which one of these helps determine the concentration of a protein by measuring its enzymatic activity? Yeah. C, yeah, an activity assay. So a lot of times when we're doing activity assays, when we're measuring the protein concentration, we'll use UV-Vis spectrophotometry to actually measure for example, the depletion of a substrate as the enzyme uses up the substrate. So you could have sort of also said D here, but what you're doing is an activity assay, and then within the activity assay, you happen to often also be using UV-Vis. All right, so finally number 4. Which one of these determines the concentration of a protein by measuring antibody binding? Yeah, ELISA, yeah, process of elimination. So yeah, ELISA is a really good, really specific technique that uses antibodies that recognize only your protein of interest, and will often use a color change at the end of an enzymatic reaction in the course of running your ELISA. So in a sense, you could also have said, oh yeah, you do use UV-Vis for this, and but yeah. But overall, this whole process of using antibodies to determine how much protein you have is an ELISA, and we'll do an example of that in a little bit also. All right, so let's compare and contrast. So let's think about gel electrophoresis as compared to gel filtration chromatography, and think about the different properties of those, how they separate whatever you're separating, what they're often used for, and which species are going to move to the bottom first. I'll give everybody a couple minutes. Feel free to work with your neighbors or your friends nearby, and we'll go over it together in a couple minutes.
Yeah. One smaller, one really small. Two useful. But God,
Yeah, so by convention, when you get a picture of a gel, the top is where you look at samples, and then the bottom is the direction they were on. Yes, that is a convention. Yeah. Yep.
So at this final step, where you're doing like a gel electrophoresis, would you use like your whole sample or just like one sample?
Yeah, so usually you just want to use a little bit of your sample because you can't recover it after you do it. Especially for protein, you're not going to recover your protein from the gel after. So you'd usually take a small amount, run on a gel, see what's in your mixture, but the bulk of your mixture you would save it in the fridge or the freezer.
Yeah. So, but for SDS page.
Yeah.
Oh, well, alright, yeah, that makes sense. Like for SDS page, you're destroying it anyway.
Exactly. Yeah. So you can't get your protein back. You don't want to put too much of it in there. Yeah.
I wasn't sure if you could like recover it or not. I only did like one gel electrophoresis in like bio one.
Yeah. Yeah. So for DNA gels, it's fairly common to recover the DNA from the gel, depending on what you're doing. Like, so for example, like if you ran PCR and you want to isolate out just your PCR product, you'll often run it through a gel, cut out your band that you want, and then isolate it from the gel. That's not uncommon. Yeah. But for proteins, that's not really done. Like you said, partially because you're sort of destroying your protein in the process of running it through the gel. Yeah. All right, so let's start going over this one. So who wants to talk about what each of these techniques is separating by? What does gel electrophoresis separate by? Yeah. Size, yeah. So gel electrophoresis primarily is separating molecules by their size. It could potentially also be using shape and charge, but because of the the environment in which we run gel electrophoresis, the shape and the charge don't matter. So in the case of proteins with SDS-Page, we are adding a detergent called SDF that unfolds our protein, so the shape is no longer relevant. And we're also adding reducing agents to break any disulfide bonds in our protein, so our proteins are very linear. And in addition, because of the detergent, they become uniformly negatively charged. So the size and the shape don't matter. Or sorry, the size matters, the shape does not, the charge does not for an SDS-Page. There are other techniques for running gels. For example, you can run a gel in native conditions that would actually take shape and charge into account, but so always make sure you're talking about SDS-Page when you're trying to talk about SDS-Page. So for gel filtration chromatography, what is it separating by? Yeah. Yeah, it's also separating by size, exactly. It can a little bit take shape into account as well. So if you think about those beads that have the little maze inside, if you have like a large protein that's like very long and narrow, it could maybe fit inside the bead. But if you have, you know, a large protein that's it's more spherical, it won't fit inside. So the shape a little bit matters. But it's mostly size for both of them. All right, so what are they used for? Yeah.
Gel electrophoresis for pure assessment.
Yeah, exactly. So it's for visualizing the results of your purification or assessing purity. What is gel filtration chromatography used for? Yeah. Yeah, it's the actual process by which you're purifying them. Yeah. So if you were going to use both, you'd probably first do gel filtration chromatography to separate out your proteins, and then you could do gel electrophoresis to see if you actually separated them the way you thought you did. All right, so let's talk about the last one. Which types of things move to the bottom first? Yeah.
For gel electrophoresis, the smaller particles are at the bottom.
Yes. Mm-hmm. And what about for gel filtration chromatography? Okay. Anybody for gel filtration? Yeah.
Largest.
Yeah, exactly. The largest ones. All right. Any questions about similarities and differences between gel electrophoresis and gel filtration chromatography, which is also known as size exclusion? All right, so let's talk about our next purity assessment method with Beer's law. So What does Beer's law demonstrate? A relationship between what two properties? Yeah. Yeah, exactly. The absorbance. So we take advantage of this relationship between absorbance and concentration because absorbance is something you can easily measure using UV vis spectrophotometry. And if you can correspond the absorbance that you measure with the actual concentration of a protein, that's pretty handy. So that's why we have Beer's law. So we have Beer's law, it's this equation, right? So what does each variable represent in our Beer's law equation? Anybody can pick any one to start with. Yeah. A is for absorbance, yes.
Is epsilon a big constant?
Yes, epsilon is a constant. It is usually called the extinction coefficient. So epsilon is a property of any particular protein. It depends on the size of the protein and also what specific amino acids are in the protein. Yeah. Yeah, every protein has its own epsilon. Yeah. All right, what about our other variables? Yeah. C is concentration. Yeah. And what about L?
For Lucas.
Yeah. If only. We all wish we could have an equation with our own name in it. Anybody got L?
Length, right?
It is length, yes. So L refers to the path length, and that's the distance light is traveling inside the little cuvette where you have your sample. It's usually 1 centimeter. Cuvettes are usually standard sizes. That way they fit inside the little UV vis machine, and they're usually 1 centimeter long. But not always, so check when you're doing this yourself. All right, any questions about Beer's law in general? All right. So let's do a practice problem. So we have here a pure sample of deoxyhemoglobin. It has an absorbance at 280 nanometers of 0.49. So if the extinction coefficient for deoxyhemoglobin is 118,872 inverse molar inverse centimeters, and the path length of the cuvette is 1 centimeter, what is the concentration of deoxyhemoglobin? So everybody take a few minutes and think about that.
use it one a lot, like a lot of techniques, or do they like continue throughout? Because like, yeah, because at the first I use high school on like halfway to Sept.
Yes, we're definitely going to get into that in later chapters. There's a variety going through. I guess it may get less technique heavy as we go forward. I think it sort of like depends on like some concepts more so, some concepts less so. But yeah, we'll go through a whole variety of stuff. Metabolism type stuff with sugars and lipids and proteins, things like signaling pathways, a bunch of stuff. Alright, so it looks like most everyone is done writing. So to solve this problem, first we start with Beer's law, right? So we have absorbance equals epsilon times C times L. So which one are we trying to solve for in this problem? C, yeah. So we can rearrange this equation to solve for C. So we can divide both sides by epsilon and L, and then we end up with C equals A over epsilon times L. And then from there, we just plug in, right? So what's our A? Yeah. Yeah, A is 0.49. Absorbance does not have any units. Our epsilon is, it's the big long number, 118,872 inverse molar, inverse centimeters. And what's our L? 1 centimeter, thank you. So if you plug that all in, the centimeters cancel out, and we get about 4.12 times 10 to the minus 6 molar for our concentration. Did anybody get a different answer?
Yeah, I used 280 nanometers and converted that to centimeters.
So. Okay, that's creative.
Yeah.
So remember L is the path length, not the wavelength of light that we're measuring. So the absorbance we're talking about is the absorbance at that wavelength, at the 280. So that's why you need to know that it's 280. But yeah, it's a very tiny cuvette, exactly right. It's an invisible cuvette. And the reason we measure at 280, does anybody know which amino acids absorb strongly at 280? Yeah, I heard it. One of them is tryptophan. And then the other one is tyrosine. So, 5%. Yeah, there's 20 amino acids. Yeah. But you could have guessed the other one. That's true. Yeah, so those are the two amino acids that absorb strongly at 280, and that's why we measure absorbance at 280, and that's why different proteins have different extinction coefficients, because different proteins have different ratios of tryptophan and tyrosine compared to other amino acids. So every protein is special. So back to our actual answer. So we had 4.12 times 10 to the minus 6 molar. Another way you might choose to write that, since we're at 10 to the minus 6, 10 to the minus 6 molar is also the same as micromolar, right? So you could have also written this as 4.12 micromolar. Sorry, my micros look a lot like little m's. It's a mu. Any questions about that? All right. So for this example, we had a pure sample, right? A pure sample of deoxyhemoglobin. What would have happened if our sample was impure? What would our results have looked like or told us?
That we had like higher absorbance, right?
Yeah, so if you have a variety of proteins, some of the proteins in the mixture are deoxyhemoglobin, some of them are not. So you might, if there was this much deoxyhemoglobin plus others, you would get maybe a larger reading of absorbance because there's like other proteins in here dragging up your absorbance at that wavelength. And also, if your sample is impure, you don't know which extinction coefficient to use, right? So if you have one protein, it has one extinction coefficient. If you have a variety of proteins, you have a problem. So Beer's law is really best applied when you have a pure sample of protein. Otherwise, your results will be inaccurate. Any questions about that? Okay. So, whoops, I forgot to show you that. Okay, so let's now talk about, sorry, this was just what I asked you, which is how would your interpretation change if it was impure? So let's talk about the Bradford assay. So first we'll just talk a little bit about what it does or what changes we're observing. So in the presence of proteins, what happens to Bradford reagent? It turns from what to what? I'll give you a hint, it's colors. What colors does it turn?
Brown.
Yes, it turns from brown to blue. Excellent. Whoops. And how do we measure that degree of color change? What type of instrument do we use? Yeah, UV visible spectroscopy. So yeah, so colors are in the visible region, right? So we can use UV vis to tell us how much of a particular color we have. So how do we then use this degree of color change that we're measuring to actually calculate our protein concentration? What do we need to measure aside from just our unknown sample? Yeah, exactly. So you need to create a standard curve. If I could spell that, that would be great. Yeah, so we need to create a series of standards that have known protein concentrations. Then you can use UV-Vis to measure how blue each of your standards is. And then when you measure your unknown protein, you can then use your knowledge of how blue it is to work backwards to figure out how much protein is in there. Any questions about that? All right, let's do a practice problem. So on the right, we have a standard curve. So those 5 different plotted points represent different known protein concentrations that we then used the Bradford reagent on and measured the blue color. So using this standard curve, which has this equation for a line, calculate the concentration of a sample, an unknown sample that had an absorbance of 0.73. So I'll give everybody a few minutes to think about that one.
Okay, so it's not the difference between the velocity itself and x. What's the zero versus
Okay, so how would we start solving for this? What equation do we want to use? Yeah.
y equals 0.13 up to x.
Yeah, exactly. So we're going to use the line, the equation that we got from creating our standard curve as our starting point. So we have y equals 0.1302x plus 0.0774. So what is our x and our y in this equation? Yeah.
Y is absorbance and x is concentration.
Exactly. Yeah, so this, just like on our graph, the y is going to give us the absorbance, x is going to tell us our protein concentration. So in this case, our absorbance is 0.73. 0.73 is our y. So now we have to figure out what is our x. So we can arrange the equation, subtract out the b, divide by the m. So we have, oops, have,
The Bradford say would be used kind of just to see the concentration of any proteins in a solution.
yeah, exactly. Yeah, we'll chat a little bit more about that once we finish the math part. But yeah, so you have y minus 0.0774 divided by 0.1302 equals x. And we know that y equals 0.73. So who has a number for x? Yeah. Yeah, exactly. So it's about 5.01 milligrams per milliliter. How did you know that those were the units? Exactly, yeah. So it's the same units as whatever your standards were in. That's what x is. The units of x, in this case, are milligrams per milliliter. It's a pretty common unit for protein concentration. And what else did I want to say about that? Yeah, oh, and you can also just visually confirm this by sort of plotting it out. So we know our absorbance is about 0.73. That's around here. So if we kind of extended it out along our standard curve, that's about here. So that's somewhere about 5. So helps if your lines are a little stronger than mine, but you get the idea. So yeah, so like one of our students just said, the Bradford assay will tell you all of the protein in your solution. It doesn't differentiate between different proteins. If you have a solution with a mixture of proteins, it'll tell you total protein concentration. Doesn't matter which proteins are in there. It doesn't tell you about specifically your protein of interest unless you have a pure solution that only has your protein of interest in it. Did anybody notice anything a little off about the shape of our standard curve? So we have this line of best fit here. Are our standards actually super linear looking? I'm seeing some head shaking. So no, right? The beginning of this curve is pretty linear. It kind of goes, pick a different color. It kind of goes pretty linear here, but our top standard is a little bit off, right? It's sort of apparently low compared to like where this line would have gone. And one reason for that is that the absorbance measured is above 1. So once you start measuring above an absorbance of 1 for any sample, you will get some inaccuracy in measurement. There's just when the proteins are highly concentrated like that, it's very difficult to get an accurate measurement. And so in this case, because our standard was a little bit inaccurate, it sort of measured an apparently maybe lower absorbance than it should have had. It can actually potentially mess up with all of our calculations downstream, because now maybe our line of best fit is a little bit off. So something to be aware of when you're doing a Bradford assay is to look at those actual absorbance values and try to get your absorbances below 1. One way you can do that, if you have a sample that's too concentrated, is you can dilute it and remeasure it. And then you can calculate sort of back from there. So in this case, this is the standard curve you have. That's what you're going to use to calculate any unknowns. But just keep in mind that it might be a little bit off, especially for higher concentrations. Any questions about Bradford assay? All right. So let's talk about an activity assay. So what type of proteins can be quantified using an activity assay? Enzymes. Yeah, I heard a couple people say it. Yeah, so activity assays are used to often measure the consumption of a substrate by an enzyme or the production of a product by an enzyme. It's measuring the activity of the enzyme, whether or not it's working. So if your protein of interest is not an enzyme, you can't do an activity assay. So let's do an activity assay practice problem. So some of you who are in the lab class might be familiar with this reaction, which is the reaction that's carried out by the enzyme LDH, lactate dehydrogenase. So this reaction that we see, we have NADH and pyruvate that are converted into NAD plus and lactate. And we're measuring this, we're monitoring the reaction at 340 nanometers. So what are we monitoring when we're looking at the absorbance at 340 nanometers? Are we looking at something getting consumed or something being produced by our enzyme? Yeah, why do you say consumed?
Because of the equation that they're using NAD as a product, and if you have NAD on the curve, it's nothing that's being absorbed there.
Right, yeah. So if we're looking at 340 nanometers, NADH, which is the dashed line here, absorbs pretty strongly at 340, compared to NAD+, which is the solid line, has no absorbance at 340. So if we're just measuring what's happening at 340, we're only going to be looking at what's going on with NADH. And we know that NADH is one of our substrates for our enzyme. So NADH is going to get used up over the course of the reaction. So as we keep monitoring our wavelength at 340, this curve might start up here at this absorbance and then slowly kind of go down as we use up our NADH. Does that kind of make sense for everybody? Okay. So what would happen if we, instead of measuring at 340, if we measured it at 260. Would this be useful or not useful to measure?
I would say it wouldn't be useful because they do absorb at different wavelengths, kind of, but it's within like a standard error pretty much.
Right, yeah, so both our product and our reactant absorb very strongly at 260 over here. So if we're consuming NADH, our absorbance wouldn't go down here because when we're using up NADH, we're making NAD. So we would still see a really strong absorbance here at 260. So we would not be able to measure the consumption or the production of either one of the products there because they will both give you absorbance at that wavelength, at 260. So we want to measure instead at 340 because only NADH has absorbance there. Does that kind of make sense for everybody? So like if you're using up NADH and you're making NAD, this absorbance here would stay fairly constant, even though the reaction's happening, because both your product and your reactant absorb there. Any questions about that? All right, so let's do a practice problem. So which sample below has the largest concentration of LDH, which is the enzyme that's catalyzing this reaction? Yeah, I heard it. So sample 3. And why does sample 3 have the most LDH? Yeah, exactly. So on our y-axis, we're measuring our absorbance at 340 nanometers. And we know, based on our picture from the last graph, as we consume NADH, the absorbance at 340 is going to go down, because if there's less NADH there, you get less absorbance. So the faster the absorbance at 340 decreases over time, the more enzyme is present to consume the NADH, right? So sample 3 has the steepest slope. This one that I'm not tracing very well has the steepest slope. So that means you have the most enzyme there that's consuming the reactant the fastest. Any questions on that? Okay, so what would happen if there was an impurity? So you had like a mixture of proteins. You had some LDH, but also some other proteins. Would that affect our activity assay?
So if you take a sample from the same thing, like the same container, then maybe not. But in theory, if you took it from 3 different ones, the absorbance would be different. So it could.
Okay, so you're saying like if you started at say at like a different absorbance because you had a different concentration.
Yeah. So if you had like 3 different samples all from the same one, then
Yeah.
maybe not. You still see like a pretty consistent. Maybe not.
Okay, yeah, let me rephrase my question. Maybe I didn't state it well. So what I mean is like if you have a tube and you have some of your protein of interest, but you also have a mixture of other proteins in it, would that behave the same in an activity assay or differently than if you had a tube with only your protein of interest in it? So do impurities affect the activity?
Well, as long as the enzyme is specific to the protein of interest, I don't think
Yeah, exactly right. So if you have LDH plus stuff, or if you have just LDH, the LDH will behave the same. It's going to consume NADH the same way. The one exception would be is if you had like an inhibitor to your enzyme in the mixture, then it would affect it, right? But otherwise, as long as your other proteins aren't messing with your protein of interest, your enzyme, then your activity assay is only measuring how much of your enzyme of interest. It's not measuring the other proteins because they're not eating up your substrates or making your products. Any other questions about activity assay? All right. Yeah.
So like the code you carry for like interacting with LDH, that would be the information or
Yeah, so LDH has a certain, intrinsic amount of speed at which it wants to eat up NADH, right? So if you have LDH and you also have, I don't know, some other random protein, that's not going to affect how fast the LDH is able to consume NADH, right? So it won't affect the activity assay. But if you had a mixture of LDH and some protein that, for whatever reason, stops LDH from eating up NADH, that would be a problem, right? That would cause it to consume NADH slower, and so it would give you an appearance of having less LDH in your sample than you actually have. But most of the time, you don't have to worry about that. Most of the time, if you have a mixture of proteins, it's unlikely that you happen to have like a specific inhibitor to your enzyme of interest. Yeah, but something to keep in mind, I guess. All right. And I guess as an add-on to that, it could also be if you had some kind of protein in your mixture that would degrade your protein of interest. Like if you just had a protease that's going to chop up your LDH, then obviously your LDH doesn't work because you chopped it into pieces. So also something else to worry about. But okay, so let's move on to ELISAs. So take a couple minutes and put in order the steps for how you prepare a sandwich ELISA. All right, so which one of these steps is first? B, yeah. So an ELISA always starts with a solid support, and the first thing you want to do is tether something directly to that solid support. So I'll try to draw a picture as best as I can. So here's our solid support. It's often a 96 well plate. It's made of plastic. It can be something else as well. And then we're going to have an antibody tethered to that solid support. So here I'll draw an antibody. Maybe the antibody recognizes little round things. I won't draw too many of them because that'll take forever. Plus my drawings are not great. All right, so that's part B, right? We have our antibody. It's red. It bound to our solid support. So what is the next step? Yeah.
Is it A?
It is not yet. Yeah. What comes before A? Yeah. C. Yeah. So in part C, we're going to then add our protein of interest. So our protein of interest is going to be incubated with that solid support. The antibody that we have tethered is going to bind to our protein of interest. So here we have our protein of interest. It has these little round things that like to stick to the antibody. So something important here is that you want to have an excess of your antibody compared to your protein of interest, because you want all of the protein that you add to be able to stick to something. You don't want to, if you end up having excess of your protein of interest, you'll end up washing away some and you won't be able to measure it, right? So you want to make sure all the protein you add is measurable. And the way to make sure that happens is you have an excess of that first antibody. All right, so what do we do next? Yeah. Then it's A. Yeah, exactly. So it is an antibody. Yeah, so this is a sandwich ELISA, and a sandwich ELISA uses two different antibodies. You might be familiar with different ELISA techniques that they go in a different order. But for the purposes of our class, we're just covering the sandwich ELISA, which is this method. So so we have step A next. We have our enzyme-linked antibody that's also going to recognize our protein of interest. So we'll incubate those together. So now we'll have our other antibody. It likes our protein of interest, so it's going to stick to it. Not like that. And here we also want to make sure we have an excess of this antibody compared to our protein of interest. If we don't cover all of our protein of interest with this enzyme-linked antibody, we won't be able to measure it, right? So we want to make sure we have more than enough of our antibody, both of our antibodies, compared to our protein of interest. And then we wash away any extra that doesn't stick to the protein of interest. So our antibody has a little enzyme linked to it. So then what is our last step? You can probably guess that it is D, right? So in part D, we're going to measure our enzyme activity, and that will help us determine the concentration of our protein. So we usually measure the enzyme activity by adding a substrate, and then often that will cause a color change. So again, we want to have an excess. We'll wash away any extra, but we want to make sure all of the enzymes have plenty of substrate so they can react to cause a color change. And the more of these sandwiches that you've created, the more color change, the more intensity of color you'll observe. And then to actually measure the intensity of the color, what type of instrument or technique are we going to use? How do we measure colors? UV vis, yeah, exactly. And so essentially the reaction that we're seeing when we add the substrate to this enzyme, the faster or the more reaction that we observe means that we have more secondary antibody that's bound, which means by because they're related, that we have more protein of interest found. So more color equals more of our enzyme linked antibody, which means we have more of our protein of interest, and that's quantitative. We can get a number from that. Any questions about ELISA? All right, so let's try to get through one more of these. So let's compare our different techniques that we just reviewed, and just say whether each one is going to be able to detect just our protein of interest, or if it contains all of the, or if it measures all of the proteins in a mixture. So our first one, our absorbance at 280, is that specific for your protein of interest, or does it just measure proteins? Anybody? Yeah, it measures any proteins, right? It's only useful when you know the extinction coefficient for your specific protein, but it's not only looking for one protein. It's just measuring absorbance at 280, which all proteins do to some extent. What about number 2? What about ELISAs? Are they specific just for your protein, or do they measure all the proteins? So when you're doing an ELISA, you're using antibodies, right? Does the antibody recognize just your protein, or is it sticking to any old protein? It sticks to your protein, yeah. So ELISA is useful for measuring your protein of interest. What about activity assay? Is it measuring any old protein or just your protein of interest? Yeah, it's just your protein of interest because you're looking at its ability to catalyze a specific reaction. How about Bradford assay? Yeah, Bradford assay is measuring all proteins. It's using a color change of the Bradford reagent, which indiscriminately sticks to all proteins. The more color you get, the more protein you have. Doesn't matter what protein it is. How about gel electrophoresis? Is it measuring all the proteins, or is it just looking for one specific protein? Yeah, so gel electrophoresis doesn't discriminate, right? It's you put proteins on a gel, it'll separate them all out so you can see how big they all are, but you're seeing all the proteins that are in your mixture. All right, so I think that's a good place to leave it for today. If anyone has any questions, I have office hours right after this. So Monday we'll finish up chapter 5, and hopefully we'll have time for some reviews. So if you have any review questions, you can bring them with you on Monday. And don't forget your quiz 4 is due tonight, and your exam 1 is a week from today. Sorry. Terrible.